Welcome to the deep dive. We're tackling, well, maybe the big question today. What actually is life?
It seems like it should be obvious, right?
Totally. Like, a rock isn't alive, a bird is. Simple. But nailing down a definition that covers everything from, I don't know, bacteria to blue whales, that's surprisingly hard.
It really is. It's something scientists, philosophers, thinkers have wrestled with forever. And instead of trying to force one perfect definition, our main source today, drawing a lot from the Nobel laureate Paul Nurse, takes a different angle.
Okay.
He suggests we try to understand life not by defining it head-on, but by looking at it through 5 key biological ideas, foundational concepts.
Right, like 5 pillars holding up the whole idea of life. And for this deep dive, we're aiming to unpack the first 2 of those pillars, see what they tell us about this huge question.
Exactly. So Nurse outlines these 5: the cell, the gene, then evolution by natural selection, life is chemistry, and finally, life is information.
And today, it's just those first 2 we're digging into, the cell and the gene.
That's the plan.
Okay, let's start there then. Pillar 1, the cell. Nurse calls this the basic unit of life. The simplest thing you can point to and say, yep, definitely alive.
Yeah, and he uses this great analogy. He calls the cell life's atom.
Life's atom.
Think about physics, right? Atoms are the basic building blocks of matter. Nurse is saying the cell plays that same fundamental role in biology. Everything bigger, tissues, organs, it's all built up from cells.
And these tiny things are, well, powerhouses. They do all the stuff we think of as living. They grow, use energy, react to things, and crucially, they make more of themselves. It all happens right there.
Absolutely. And, the story of figuring this out goes way back. 1665. Robert Hooke.
Oh, with the microscope?
Exactly. One of the earliest microscopes. He looks at cork, sees these little boxy compartments, and they reminded him of the small rooms, the cells, in a monastery. So that's the name he gave them.
Monastery cells.
Yep. And he published it all with these amazing drawings in his book, Micrographia.
Wow. And that name just stuck. Amazing. Paul Nurse even talks about seeing cells himself, right? Plant root cells.
He does. He shares this personal moment looking through a microscope early on and seeing these fundamental units.
Yeah.
He said it was just this moment of clarity, really set him on his path.
Yeah, you can imagine that.
And, Nurse really brings it home for you, the listener. He points out that every single person listening, every human, started life as just one single fertilized cell.
Whoa.
It's pretty profound. A reminder of how connected all life is, starting from that one basic block.
Okay, that really sets the stage for the cell. Now, connecting that to Nurse's own work, that brings us straight to pillar number 2, the gene. Because his research was all about how cells divide, right? And he used yeast.
Yeast, yeah.
Yeah.
Seems humble, doesn't it? Bread and beer.
Right.
But Nurse explains why it's, such a brilliant model organism. It's simple, but its cells are eukaryotic. That means they have a nucleus, complex insides, like our cells do, so it's relevant. Plus, they grow fast, easy to handle in the lab, and scientists have, amazing genetic tools to work with them now.
So looking at yeast dividing could actually tell him about how our cells divide.
Precisely. And this led to just a stunning discovery by his team. They found a specific gene in yeast that controls cell division. But then, and this is the kicker they found, you could take the human version of that same gene.
The human version?
Yeah. Put it into the yeast cell that was missing its own copy, and the yeast cell divided perfectly normally.
Hold on. You swap in a human gene, and the yeast cell is fine. It just carries on dividing. That sounds, how is that possible? We're so different.
It seems completely counterintuitive, doesn't it? And that's why the discovery was so huge.
Okay.
Its significance is, it shows that the basic machinery for cell division is unbelievably ancient. It's been conserved, kept almost the same, across these vast stretches of evolutionary time.
How ancient are we talking?
This core process likely emerged over 1.5 billion years ago, long before animals, before plants.
Yeah.
Way back in our single-celled ancestors.
1.5 billion years. And the gene still works in yeast and in us? Wow. What does that fundamentally tell us, that something so critical has stayed the same for that long?
That's the really deep insight, I think. It suggests that life figured out some really essential molecular tools very, very early on. And these tools were so good, so vital, that evolution just hasn't messed with them much. It's like, Nurse uses this analogy, it's like you could take a piston out of a Maserati and swap it into an old Ford, and it still works. Except here, the brands are separated by, eons, not just decades. It shows this incredible shared heritage at the molecular level.
Okay, yeah. The Maserati-Ford comparison really hammers home just how conserved that is. Right, so that's the gene in action shown by Nurse's work. But stepping back, the idea of a gene, where did that even originate? People knew things were inherited long before DNA, right?
Oh, absolutely. The concept came way before we knew what genes were physically made of. And for that story, you have to go back to the 19th century. Gregor Mendel.
Ah, Mendel, the monk with the pea plants, yeah.
That's the one. Gregor Mendel. Monk, scientist. And his real genius was his method. See, before him, people just kind of described inheritance. Tall parents sometimes have short kids, etc.
Right.
But Mendel, maybe because he'd studied physics and maths, he took a rigorously quantitative approach. He didn't just observe, he counted.
He was literally counting the peas or the plants.
Exactly. He did thousands of controlled experiments crossing pea plants, tracking specific traits, height, flower color, seed shape, you name it. And by meticulously counting the offspring in each generation, he noticed these incredibly consistent mathematical patterns.
The famous ratios.
The famous ratios, like the 3-to-1 ratio when you cross two different purebreds and look at the next generation,
Yeah.
or the 9-to-3 to 3-to-1 ratio if you track two traits at once.
And purely from those numbers, with zero knowledge of DNA or chromosomes, he proposed that traits weren't just blending, they were controlled by distinct hereditary units.
That was the conceptual breakthrough. He called them factors or units. He had no idea what they were physically, but the mathematics demanded they exist as discrete things passed down. He basically figured out the rules of gene behavior just by careful observation and counting.
So he laid all the groundwork.
Absolutely. Defined the basic unit of heredity, setting the stage for everything we know about genetics today, even though the physical gene itself remained a mystery for decades.
Okay, so in this deep dive, following Nurse's framework, we've looked at the cell life's fundamental unit, from Hooke's discovery onwards and the unit of heredity, from Mendel's conceptual leap right up to Nurse, showing these ancient, shared genetic mechanisms with his yeast work.
And like Nurse says, these are just the first two steps. His framework also includes, the driving force of evolution by natural selection, the complex processes of life as chemistry, and the crucial role of life as information.
Right, lots more to explore there in future discussions, definitely.
For sure. But for now, we've seen how these first two pillars, the cell is the basic building block, the gene is the instruction manual, give us this picture of life built on incredibly ancient shared foundations. That gene swatching story really drives that home.
Yeah, it really does. And Nurse's way of weaving together the history, his own research, those analogies like life's atom or the engine parts, it makes these huge ideas feel much more, graspable. It definitely offers a powerful lens on that what is life question.
It does. And when you think about how deep that conservation runs, these fundamental processes working across billions of years in bacteria, yeast, plants, us, it really highlights this profound interconnectedness, this shared history.
So thinking about that deep shared history, that ancient biological machinery we all carry, what does that really tell us about how connected all life truly is? And maybe, how does viewing life through that lens, seeing it as this ancient system running on conserved chemistry and information flow, how might that shift our whole perspective on ourselves, on other creatures, on our place in it all? Something to think about until next time.